Good morning, good afternoon, good evening, wherever you are on this planet, and welcome to our SETI Live. My name is Frank Marchis. I'm a senior researcher and director of citizen science for Unistellar at the SETI Institute. Today, we are going to talk about Mars, and specifically, we are going to talk about the fact that we have been searching for life on Mars for decades, and maybe we have been looking for life in the wrong place. And to talk about this, because I'm not very focused on Mars, we invited Dirk Schulze-Makuch. Hi, Dirk, how are you?
Hi, how are you doing? Thank you for having me.
So, Dirk, you are professor at the Center for Astronomy and Astrophysics at the University Technical of Berlin in Germany. That's where you are calling us from, I'm assuming?
Yes, that's correct.
Okay, and you're also adjunct professor at the School of Earth and Environmental Sciences at Washington State University. I look at your Wikipedia page and I saw you have been co-author on 5 books on astrobiology, and I recommend our viewers to check out. There is some very interesting book with very cool topics. I will love to read them. That's one of the things I will do in the next summer break. But today we are not going to talk about all your books. We are going to talk about this comment article you published recently in Nature Astronomy entitled, "We May Be Looking for Martian Life in the Wrong Place." So, wrong place and Mars. Before we go to the research, you've been working on this field for quite a while. You know a lot about Mars. You know a lot about this planet. And you know that there is some people who are gonna send maybe one day humans on this planet. So let's assume you go on Mars. We give you a ticket to fly and to land on Mars. Where will you go?
Well, what I would really like to see would be standing on top of Valles Marineris and look in this huge canyon down there. But, in order to look for life, I would probably go to the Southern Highlands on Mars and look there and take my hammer with me and look at some of the salt rocks and see whether I can find life in them.
Okay, so you will not go on Mars for the view. You will go to look for life.
Preferably both.
Okay, good. All right, good. We're gonna talk about that in more details. All right. So yeah, let us know where you're watching us from. I saw there is some people joining us from Sweden, Pakistan today. But let's talk about the science. So, can you give us a very short summary of the core idea behind your paper?
Well, the core idea is that from the research that we do in the Atacama Desert, which is an analog for Mars, uh, it is hyper-arid, so it's very, very dry. And the organisms are adapted there to that dryness. They will not have any liquid water from rain or so, but just from the atmosphere. That's called hygroscopicity. So they take the water directly from the atmosphere. So they are adapted to this very, very little water. Now, if you have this adaptation and the same kind of adaptation we would expect on Mars, and put too much water on it, you flood them with water, you kill them, you basically drown them. And I think that might have happened during the Viking experiments, actually, and this kind of strange kind of result that we got out of it. So if you want to search for life on a planet like Mars or any planet, you really have to be aware of the environment and then proceed accordingly.
Okay. Yeah, so you mentioned the Viking landers had experiment to search for life. He landed 50 years ago, roughly. So basically, your paper say, well, we could not have detected life with the Viking experiments because these microbes, if they exist, they're so sensitive to water that they have been killed by the experiment itself. Is that correct?
Well, yeah, in principle, that is correct. I mean, the thing is, there are several things going on at the same time. The labeled release experiment was conducted with water, and I mean, you had some positive results, but then suddenly they turned negative, and you didn't know what really was going on. And I think with that, the water issue could have resulted in those confusing results. the pyrolytic release experiment that tested for organic synthesis. The first one was actually positive by 3 sigma, by 3 standard deviations. And the other experiment that followed, the worst results was the only experiment that was done under wet conditions. So there is a really telltale signs in the Viking experiments. And since those are the only life detection experiments ever conducted on another planet, I mean, we should really have another look at it and look from our experience 50 years after, when we have a better ideas about the technique and also about the Martian environment.
Yeah. All right. And this kind of idea doesn't come from the thin air, no pun intended here. It comes from the fact that people have been visiting extreme environment on Earth, like very dry desertic area, like the Atacama Desert, for instance, and they found some similarities between Mars environment and this dry environment. Is that correct? What kind of life have we found on the desert of Atacama?
Right, there's kind of an ecological sequence that you see to dry and dry environments. And the last life, basically, that you find, if it's extremely dry, it is life living in those salt rocks that use thin air, as you say, or the moisture in the air really to get their water from. And if it's so, if that salt that we're talking about is so hygroscopic that it produces actually a brine, a liquid state, then we call it the liquescence, and then this is even more extreme. And by this process, the microbes get their water, and by drying out, there could be the water activity on the microscopic scale could be still high enough, even on Mars, to make a living.
Okay. Let me say hi to some viewers joining us from various places around the world, but from Rio Grande do Sul in Brazil, in California, Louisiana in the United States, Florida in Brazil, North Carolina. Welcome. We are talking about a new hypothesis on the search for life on Mars, with the idea of following the salts to find life, which I like that. In fact, we all remember that one of the motto of NASA was follow the water. And now you basically propose, Dirk, that we should be not following the water if we want to find life on Mars, but we should be following the salts. So you talk about hygroscopic salts. So those are water attracting salt, right? That's basically, it's a salt for which water is being captured. So how's this water? Can you tell us a little bit more details about this kind of salts?
Well, there's different type of salts that are hygroscopic, and even the table salt that you use in the morning, maybe, in California it's 10 a.m., you probably just had your egg, right? The breakfast.
Yeah.
And if you look at the salt dispenser, a lot of the salt dispenser, especially in the restaurants, they have rice grains with the salt. Why is that? Because rice grains are even more hygroscopic than the salt. If you just leave the salt out, it's getting clumpy. So even the normal table salt, sodium chloride, is hygroscopic and would be working for those microbes. But then we also have other salts, for example, perchlorates. Of course, perchlorates have some other disadvantages as well, but also advantages. And we found a lot of them, or a high concentration of them on Mars. So those salts are possibilities. And there's quite a few other salts as well.
Only with you, Dirk, we can start talking. We talk about Mars, and then we ending up talking about eggs in the breakfast. I love it. I love it. Thank you. Okay, so if you have to redo an experiment to find life on Mars, I give you funding magically, billions of dollars, right? What will you do then?
Well, I would send a mission to the southern highland of Mars, or one of the other areas where we know that we have a lot of salts. And then I would have a little, at least a scraper with me to scrape off some of the top salts, that And microbes use the top salts basically for UV radiation for to protect them from that, and go a little bit further, and then do some kind of analysis of organic compounds, if I can find organic compounds. And I, of course, want to have a microscope with me, right?
Okay.
Because one of the things that our work group actually in Berlin working to is identifying microbes by their motility. So if something is moving there and it's not following Brownian motion, so this normal stuff what sediment grains do, but have specific paths to nutrient source or away, then I think we can get a good evidence for life on Mars.
Okay. So those are microbes you will be looking for, like any type of microbes. You have something that's of some very specific type of microbes. What
Well,
will you be looking for?
that would be halophiles, of course, microbes that are adapted to high salt concentrations. But those could be really different type of microbes. We find quite often cyanobacteria as well. There are cyanobacteria that adapted to high salt conditions and live in hyperarid environments. We see in the rock sometimes this kind of greenish veneer, and but there could be others. Usually it's a kind of, in the Atacama Desert or on Earth, it is kind of a communities that work together. And if you look for life on another planet, of course, we're not looking for a single microbe. We would look for some kind of an ecosystem, even if it's a primitive or simple ecosystem that of organisms that are working together, probably not all the same ones.
Okay. So I remind our viewers we can take some questions if you want to know a bit more about Dirk research and this interesting idea of searching for salt to find life. Sorry. So I saw recently that I think that was Perseverance rover broke some kind of rock, and this revealed some very diversity in color of the inner side of the rocks. Have you seen those pictures? Do you know what I'm talking about?
Yes, I know what you're talking about. That's always fascinating if you see the rocks and the diversity. Actually, that reminded me of some rocks that I took on the Azores near hydrothermal area, and they were all colorful. And within about 10 centimeters or 4 inches, there was 5 different beautiful colors. We took samples from each of them and found in each of them really different kind of microbes. I mean,
okay.
whether this is there on Mars is a different question, right? But it is kind of intriguing to have such a rock, and you want to have it at home in your lab and look at it.
Yeah, to be certain, because colors is not enough to tell you whether or not there is microbes.
Right. Yeah.
Colors
And
can be due
texture not
to
really either. So you need more. You really need. In astrobiology, I always tell my students you have to get in close and personal.
Good. I saw we have some questions popping up. So we have one already from Virtual Space Agency. Are the current rovers capable of detecting salt?
As far as I know, yes, they are able to detect salt. But where the rovers are, that is in where were lakes before. And most of the salts are there sulfur salts, and sulfur salts are unfortunately not hygroscopic. The most salt deposits, so we know from the orbiters actually, because we can look for chloride, for example, and that's then mostly then sodium chloride. And of course, rovers detected, for example, perchlorate.
Okay, so perchlorate is a kind of salt, and we're detecting this from orbit using those satellites that map the surface of Mars in the near infrared or something like that?
No, the other way around. The perchlorate was discovered by the rovers because they are at a lower concentration. So the table salts, the sodium chloride, and so that is detected from the orbiters.
Okay. We have questions. One question is from Ray Casa. How will we detect it or recognize it? So I think it is life.
Of course, that is $1 billion question also for your mission, what we do exactly. I actually thought the Viking detection life experiments not bad designed, because there were 4 different kind of ways how to look for it, right? You had the GCMS looking for organics. You had the labeled release experiment for looking for metabolism. You had the pyrolytic system for organic synthesis. And then you had the chemical, looking for the chemistry. I think we need to have something. We cannot rely on one system only. Of course, with our sequencing ability, it would not hurt to have a sequencer with us, one of the small nanopore sequencers. But I would like to have more. I would like to have the organics there. I would like to see if there are organisms, hopefully that they're alive and we see them moving, and maybe have something to analyze the biochemistry to see the molecular makeup.
Okay. Another question for Lisangalo Berti, which is very related to what's going on at the moment. So the samples that were left in the Jezero crater by the Perseverance mission, do you think it's worthwhile to take them?
That is in a way a difficult question. Of course, we would like to have them here, but it's very expensive to get them back over here. I think the more direct way would be an in situ mission, a live detection mission, a robotic mission that goes there and does this kind of experiments and looks for life, rather than bringing samples back with a sample return. But having said that, of course, that increases still our knowledge, and certain things we cannot do in situ what we could do at home in the lab. But what I would be worried about most is that if we have a really living organism in the sample, by the time we get it back, it's stored over there, moving through space, it's dead, and we just have the remnants of it. I would like to see life living, so to speak.
So this is one of my questions, because I've been wondering this for months and even years. So I'm gonna ask a very dumb question: why did not we send a spacecraft, a rover with a microscope?
Well, that's a good question, because I'm asking that sometimes too. So, well, we did send with a microscope, but there's, for example, the atomic force microscope, but that was more suitable to look at minerals, or to look at too small scale really for microbes. We didn't send a microscope that was able to look for microbes. It is a little bit, it's also a technical issue, because the best you could do, with that, if you actually have a human that looks through a microscope over there, to do that remotely, it's a bit of a challenge. But I think we are far enough that right now that is something that really should be done.
Yeah, that's something that should be done. I think it should be the next generation of rovers should have a microscope, and
I really think so too.
Yeah. Okay, so we have some question. One question from Ron Schneider about life in that can be found deep in the caves and lava tube beneath the surface. What's your personal guess on that?
I think that is most likely that life exists there. The big problem is just to get there. I mean, if you have a lava tube cave, and lava tube caves are bigger on Mars than on Earth, and they go deep enough inside, you should have liquid water at some point. And I firmly think that there was quite a bit of microbial life on Mars like 4 billion years ago. Then the environment got very, very difficult for life, and life had to retreat back into habitats, into niches. And that would be some of the niches where I would expect life to exist. The only thing why I favor salt rocks is because they are much more accessible. because I think there we could find life actually within the upper 10 centimeter of the crust, basically, or maybe 20 centimeters down. But then we could still find life. If you go to lava tube caves or lava tubes or caves, another caves, you have to go, I would think at least 50 meters down, maybe 100 meters or more.
Yeah, that's a good point. It's possible there is life on the surface surviving in this extreme environment because of its UV and it's very dry and it's accessible. So it's like we are searching for life, but we cannot search everywhere on the planet. So the main idea here is that you giving us a place where you believe that extreme life could exist, the salt, the place where the salts are.
Right.
But it's very likely, and in fact, we had a meeting yesterday at the SETI Institute, and I forgot the name of the researcher who basically say she truly believes that there is life, like you just say, underneath the surface of Mars. The life that survived the time where Mars was hospitable with liquid water on the surface and so on. And this life retreated, as you mentioned, and went to hide basically and survive in the caves. So what we just need to do is to go there and to explore those caves.
Right. Well, that is difficult, but now maybe with drones or something, you could do it, go further. Yeah. But then, I mean, you not only want to go with the drones inside, you still, when you find something interesting, you want to analyze it. So the drone still has to carry maybe a small GCMS or something else, some kind of instrument that can do something. So
that's a good link to the question from Jason Daniels. So do we have the microscopy technology small enough to analyze the light? And oh my God, I misread the question. Then do we have microscopy technology small enough and light enough to send to Mars? So basically, can we have miniaturized microscope or miniaturized instrument that will, without any doubt, tell us that there is life on Mars?
I mean, it's definitely it's small enough and light enough to put it on a rover, like on the Curiosity rover or Perseverance rover. I mean, there can be instruments at a few kilograms, and that definitely would work.
Okay, that's an interesting view. So the future is bright for the Mars exploration, based on what you're just telling us, right?
Well, I don't know. I mean, we at least right now, I mean, the sample return mission, I think, is still on hold, temporarily on hold. Mm-hmm. So we don't have any other new mission really planned. From the European side, there's an ExoMars mission. That seems to be a go now, but of course it's delayed by quite a few years. I think it's 2029 or 2030. So, but it would be nice to have some kind of other missions. And what I really like to see is a true life detection mission, something like Viking. I think right now, 50 years after Viking, we understand the Martian environment good enough that it's really time to have it.
All right. So if there is any viewers who have some billions of dollars and want to be the first one to sponsor a mission that will find life on another planet, Dirk has some ideas and believe that Mars is the right place to go, especially where there is salt. That's I think a summary of this conversation, right?
Yeah, sure. Absolutely.
Okay. Good. So what's the next step for you beside this big project? Are you working on some other ideas to explore Mars? What's the next big thing?
The next big thing, my research group works on motility, and with artificial intelligence, basically decide if we have microscopically look at particles or microbes to move to determine, whether it's microbial or whether it's just a centimeter particle, and even to say, at least for Earth microbes, what type of microbes that actually are that are in the liquid. So we are working on that to put this kind of together. some kind of apparatus to kind of, if you have a sediment, you put some liquid on it, and then you see things are moving, and to determine what it really is. And
Okay.
I think that would be really useful for a place like Mars, but we have also application on Earth because we use it, for example, to identify pathogens like cholera or so in water.
Excellent. That's a great project. And I hope you're going to come back to talk to us about this, because I know our audience is very interesting to hear more about how we use AI to do astrobiology or to search for life elsewhere in the universe. So that would be a perfect topic. When you're ready, let us know. Okay. Well, thank you very much, Dirk. Very appreciate. I hope to see you in person soon at the SETI Institute, or maybe when I go to Berlin, which is my favorite city in Europe. But don't say that.
You're very welcome. Just let me know when you're there.
Okay. Thank you to all our viewers. This was a SETI Live by the SETI Institute. We are a non-profit organization, as you know. You can support us by liking this video, commenting. You can ask questions as well. We take the time to answer to some of them, of course. You can also join, visit our website and get our newsletter called Journée, where you will basically find out everything happening in the field of astrobiology on a monthly basis. And if you're inclined, you can also click on the donate button in our website and make a small donation to support this program, to support the research of our researcher at the SETI Institute, and maybe help developing the next miniaturized microscope that will be on board a drone to explore the cave of Mars and find life. So thank you very much. Thanks again, Dirk.
Thank you.
Goodbye.
Bye-bye.